Welcome to this explainer. Today, we're going to unlock the electrical mysteries of the simplest amino acid in existence, glycine. If you've ever found yourself wondering how biological molecules actually manage their electrical charges to survive in totally different environments, well, you're in exactly the right place. We are going to look really closely at how glycine behaves under pressure, specifically how it reacts to changing pH levels.
Our agenda today covers 5 quick stops. First, meeting the amino acid glycine. Then, understanding pKa and Pi. After that, we'll walk through the titration experiment, learn how to read the titration curve, and finally, calculate our final results. Let's get right into it.
Section 1. Meet the amino acid glycine.
Now, glycine gets a rep as the simplest amino acid, right? But honestly, its behavior is anything but basic. Glycine contains 2 highly reactive ionizable groups, a carboxyl group and an amino group. And what makes this so fascinating is that these groups act, well, kind of like chemical chameleons. Depending entirely on the pH level of the environment around them, glycine will literally shift and change into totally different ionic forms. It's a molecule with a true dual nature, and that duality is exactly what we're going to measure today.
Moving on to Section 2, Understanding pKa and Pi. You're probably familiar with the Henderson-Hausebach equation, pH equals pKa plus the log of the deprotonated molecule concentration over the protonated one. But what does that actually mean for us in practical terms? Well, the pKa represents the exact pH level where half of your molecules are protonated and the other half are deprotonated. It's this perfect 50-50 split. So, by finding that exact point, we're fundamentally measuring the acidic strength of a functional group.
And that perfectly tees us up to talk about a really fascinating chemical state called a zwitterion. Imagine a single molecule that has a positive charge on one end and a negative charge on the other, balancing out so perfectly that overall, it has absolutely no net electrical charge. That's a zwitterion. And this state occurs precisely at the isoelectric point, or the Pi. Finding this exact point of balance for glycine is basically the ultimate goal of our analysis today.
All right, Section 3, the titration experiment.
To actually see this in action, you don't need some massive, crazy lab setup. We're talking just a few key essentials. Primarily, we need our starting glycine solution, a strong base like sodium hydroxide, NaOH, to force the chemical reaction, and a highly sensitive pH meter to track the whole journey. Throw in a burette to dispense the base drop by drop, plus your standard laboratory glassware like beakers, and we're good to go.
The procedure itself, it's really an exercise in patience and precision. First, you take the glycine solution in a beaker and log its starting pH. Then, you systematically add the sodium hydroxide from the burette, making absolutely sure to continuously stir the solution. After each and every addition, you carefully record the new pH. You just continue this process over and over until the pH rises significantly. Basically, you're mapping glycine's changing chemical states in real time.
But here is the absolute crucial thing to remember before we even talk about data. Precision is everything. A brilliant experiment is completely useless without reliable data, so you have to calibrate your pH meter properly before even looking at the glycine, and you must add your sodium hydroxide slowly. If you rush it, you're going to miss those subtle chemical shifts we're trying to capture, and your final calculations will be totally off.
Section 4. Reading the titration curve. Once you actually graph those readings, the shape that emerges is incredibly revealing. You'll see 2 distinct, flattened out areas. Those are your buffering regions. That first buffering region corresponds to our first pKa, which is the carboxyl group reacting. The second region corresponds to the second pKa, the amino group reacting. Now, if you pinpoint the exact midpoint of each of those flattened buffering regions, you hit the half equivalence points. At these exact milestones, the pH of the solution equals the pKa value of that specific group.
So reading that curve gives us the hard numbers. Our experiment shows that the pKa for the carboxyl group, which we designate as pKa1, is roughly 2.3. That is highly acidic. But the pKa for the amino group, or pKa2, sits way up at approximately 9.6, which is a much more basic environment. Glycine essentially has 2 totally different chemical personalities mapped out on opposite ends of the pH scale.
And finally, Section 5, calculating the final results. Now, calculating the isoelectric point for glycine is honestly quite elegant. Because we're dealing with exactly 2 ionizable groups, finding the exact pH where the molecule has 0 net charge is as simple as finding the mathematical middle ground. We just take the average of our 2 newly discovered pKa values.
Let's crunch those numbers real quick.
You take that pKa1 of 2.3, add it to the pKa2 of 9.6, and divide by 2? Boom! That reveals the isoelectric point of glycine is approximately 5.95. At a pH of exactly 5.95, glycine just floats in the solution as a perfectly balanced zwitterion, with its positive and negative charges completely neutralizing each other. Before we completely wrap things up, I want you to think about a classic Viva question that biochemistry students almost always face. Why exactly does glycine act as a buffer? And why does it exhibit two distinct pKa values instead of just one?
The answer, as we've just seen, lies squarely in those two distinct ionizable groups, the carboxyl and the amino, that we successfully mapped today. Because they react at vastly different pH levels, they create two entirely separate buffering zones. And listen, this isn't just some abstract lab exercise. This is a foundational concept. Understanding how amino acids like glycine act as buffers helps explain the vast, complex biological buffer systems that regulate our blood, our cells, and ultimately keep us alive. Thank you so much for joining me on this explainer. Keep exploring the chemistry around you, and next time you think about the delicate balance of life, ask yourself how many perfectly balanced zwitterions are working inside you right now.